Hello everyone, welcome to the Postlab session for exercise number 3, which is membrane transport. This is in preparation for your upcoming first long exam. So let's begin. So for the first question, it was asking what is the molecular weight of hydrochloric acid with the correct units? And the correct answer is 36.46 grams per mole. You can also decide to answer it in kilograms per mole. And I would accept this as long as it is the right value, of course. Next, what is the molecular weight of NH4OH, basically ammonium hydroxide? The correct answer is 35.04 grams per mole. Now, given their molecular weights and the same time and temperature, which compound will travel faster in 15 minutes? And you are asked to provide a clear and concise explanation for your answer. So considering that NH4OH is slightly lighter than hydrochloric acid, then NH4OH would generally travel faster and farther in the same time period compared to HCl. And this is because the heavier the molecule, the more difficult it is to travel across a certain space and time. Okay. Now, what compounds are formed upon the reaction of hydrochloric acid and ammonium hydroxide? How would this be useful in trying to predict the distance traveled by HCl and NH4OH in a limited period of time? So upon meeting, NH4OH and HCl would undergo a chemical reaction that would form the compound NH4Cl. And NH4Cl is a white precipitate that is useful for predicting the distance traveled by both gases because it will allow the gases to become visible. Naturally, hydrochloric acid and ammonium hydroxide are colorless, but once you see a white cloud or a white ring form within your glass tube, you know that HCl and NH4OH have met and therefore they have traveled that distance. Next, what is the molecular weight of the following compounds? So for Congo red, it's 697 kilograms per mole. Methylene blue is 319 kilograms per mole. And potassium permanganate would be the lightest at 158 kilograms per mole. So given all that, which among them will diffuse fastest in an agar medium? And the correct answer would be potassium permanganate. And this is because it is also the lightest compound. And as mentioned earlier, the heavier the molecule, the more difficult it would be to travel in a particular space and distance. Okay? So since potassium permanganate is lighter, it can travel faster. Next, you were then asked to describe the expected distribution of potassium permanganate in each beaker described in the handout. Okay, so one was heated to 50 degrees Celsius and the other was with ice. So after a certain period of time, the potassium permanganate in the hot water would have diffused evenly across the solution, whereas the potassium permanganate in the cool or cold liquid would remain undistributed, possibly concentrated at the bottom because it would be unable to distribute itself because of the lower temperature. And this is because at higher temperatures, a system would have higher kinetic energy, which would allow the molecules in the system to move faster. And that's why at higher temperatures, diffusion occurs faster because the kinetic energy speeds up the movement of the molecules that are undergoing diffusion. At lower temperatures, since there is less energy in the system, there is less movement, and therefore the diffusion will not have happened fast. Okay? Now, why is potassium permanganate the chemical used in the diffusion and temperature experiments? Well, as mentioned before, potassium permanganate is a light molecule. So because of this, we can see the effects of the experiment at a faster duration, so it saves us time. But apart from this, potassium permanganate is also very brightly colored. So we can visually see the effects of diffusion and temperature. And then finally, potassium permanganate is also relatively safe. It is not toxic and will not endanger anyone performing the experiment. Next, in the experiment involving placing a cellulose sac with 1% starch solution in a beaker of water with drops of iodine, where should the color change reaction occur? So please take note that in this experiment, the starch was inside the cellulose sac and the iodine was in the beaker. Since starch is too large, is a large molecule, it cannot pass through the cellulose sac because the cellulose sac is a semi-permeable membrane that can only allow small molecules to pass through. As such, iodine would be able to pass through, and so it can go from the beaker into the cellulose sac, and thus the color change reaction would happen inside the sac. Next, in the experiment involving placing a cellulose sac with 1% starch solution, what is the reason for the color change reaction occurring where it did? So as I mentioned a few minutes earlier, the cellulose sac is actually a semi-permeable membrane. It only allows certain molecules to pass through. Specifically, it only allows very, very small molecules to pass through. And in this case, starch is a large molecule and iodine is a very, very small molecule. So although starch will have a tendency to diffuse out, it won't be able to do so because it cannot pass through the cellulose sac. Iodine, on the other hand, would have a tendency to diffuse into the sac, and since it's small, it will be able to pass through. And that's why the color change reaction would happen inside the cellulose sac. Now, given the following data for the measuring of the rate of osmosis experiment, we were then asked to compute for the percent change in weight. The formula for this would be final minus initial, all divided by initial times 100%. So for the first setup, you will get 0%, the second setup 10%, the third 20%, and the fourth 30%. And when it comes to the fifth, since the final weight is less than the initial weight, you will get negative 30%. So given the table in the question above, what is the relationship of the rate of osmosis with the concentration of sucrose on one side of the membrane? So as you can see, if you actually plot the change in weight of the different setups for the first four setups, you can see that the higher the concentration difference, the higher the change in weight, and thus the faster the rate of osmosis. And this is because the larger the concentration gradient, or basically the larger the inequality across a semi-permeable membrane, the faster the rate of osmosis. And this is because the system would want to achieve a state of homeostasis or balance at a faster rate with a greater degree of inequality. Okay? Now, when it comes to the results in sac number 5, it was unique because instead of the sac gaining weight, it actually lost weight. And this is because I want you guys to remember that osmosis is the movement of your solvent molecules, specifically water, from an area of low concentration to an area of high concentration. And as you can see in the fifth setup, the area of low concentration is inside the sac. It's the distilled water. Whereas the area of high concentration is in the beaker, which contains the 1M sucrose. And thus, since sucrose cannot pass through the cellulose sac because it is a semi-permeable membrane and sucrose is a large molecule, water will be the one to move. And since there's more water or there's a lower concentration in the sac, the water will have a tendency to move out, and thus the sac will become lighter over time. Okay, I repeat, this is because water moves from an area of low concentration to an area of high concentration. And since the sac has a lower concentration than the beaker, the water from the sac will move out. Okay, and thus it becomes lighter over time. Now, in what way is a cellulose sac or dialysis sac similar to the cell membrane? Well, similar to the cell membrane, the cellulose sac is semi-permeable. It only allows small molecules to pass through. And the cell membrane is the same. It can only allow small molecules to pass through also, particularly small uncharged molecules. And that's why if you have larger molecules or charged molecules passing through, it will need to be facilitated by membrane proteins, okay? And thus, it would be called facilitated diffusion if that was supposed to happen. Our cellulose sac, however, does not have these protein channels, and thus facilitated diffusion cannot happen in the cellulose sac. Now describe the shape and size of the red blood cells seen on a glass slide with 0.9% NaCl. So as you can see from this image, your red blood cells would be oval in shape, they would be healthy, and they would be normal. On the other hand, if you look at the red blood cell with distilled water, you can see that they are enlarged, they're bloated, and some of them have actually burst or lysed. And then if you compare this with the red blood cells found within the 5% NaCl solution, a lot of them have shrunk down or cremated, and this is because they are very much stressed by the environment. So nag-shrink sila. Now, based on the results of the experiment on the osmosis in the red blood cells, which solution is hyperosmotic relative to the red blood cell? So the one that is hyperosmotic or will cause movement of water out of the cell would be the 5% NaCl solution. So it has a higher concentration of salt and will thus cause the water from inside the cell to move out, thus causing the cells to shrink and cremate. The next is, which one is hypoosmotic relative to the red blood cell? And the answer for this is the distilled water. Distilled water would have less concentration of salts present in it and would thus cause water to move into the cell, causing it to bloat, to expand, and eventually to burst or to lyse. And then finally, which one is isosmotic relative to the red blood cells? And this would be your 0.9% NaCl. So this has the same concentration of salt and would thus allow your red blood cells to remain normal and healthy. Okay. So we now move on to your recitation answers. If you have any questions about your Postlab, please don't hesitate to message me via Anonymous Space and I will try to answer within the next 24 hours. Okay. So for the recitation, the first one was to differentiate diffusion from osmosis based on which molecule moves. So in the case of diffusion, it's the solute molecules that are moving, whereas in osmosis, it's the solvent that's moving, specifically water. For the second recitation question, differentiate diffusion from osmosis based on the direction of movement of molecules, if it's up or down the concentration gradient. In the case of diffusion, it moves down the concentration gradient as molecules are moving from an area of high concentration to an area of low concentration. On the other hand, for osmosis, it's going up the concentration gradient because water moves from an area of low concentration to an area of high concentration in order to achieve homeostasis. For number 3, if I place a cellulose sac containing iodine and a beaker containing starch, where would the blue-black color reaction occur? So as you can see, I inverted the experiment described in the worksheet to test if you actually understand the principle. And since the cellulose sac is again a semi-permeable membrane, iodine will be able to move, but starch will not. And since iodine is the one inside the sac and starch is the one outside, iodine will diffuse out of the sac and thus the color change reaction will happen out of the sac or basically in the beaker. Next, if I place a cellulose sac containing 0.4 m sucrose in a beaker with 1 m sucrose, will the sac be lighter or heavier after 30 minutes? So the correct answer is lighter. Because remember, sucrose will not be able to move across the cellulose sac because sucrose is a large molecule. And the cellulose sac is a semi-permeable membrane that only allows small molecules to pass through. So, osmosis will be the one to happen. So, water will move from an area of low concentration to high concentration. So, from 0.4 m to 1 m, thus causing your cellulose sac to be lighter over time. And then number 5, if I have a 10-meter glass tube and I place a cotton ball with ammonium hydroxide on one side and a cotton ball with HCl on the other hand, and after 30 seconds, a white cloud appears 4 meters from hydrochloric acid, what is the rate of diffusion of ammonium hydroxide? So the first thing you have to compute for is the distance traveled by ammonium hydroxide, which is basically 10 meters, the length of the glass tube, minus 4 meters, the distance traveled by hydrochloric acid. And so what you'll have left is 6 meters. And thus, the rate of diffusion of ammonium hydroxide would be 6 meters divided by the time elapsed, which is 30 seconds, and there you will have 0.2 liters per second. So more or less, that ends the post-lab discussion for exercise number 3. I hope this is helpful for your upcoming exam. And together with this link, I will also be sending you the image file for your first long exam, which I hope you will download ahead of time so that you already have the images with you during the test. So wishing you all the best in your preparation. Please enjoy the holidays, and I'll see you this coming Tuesday. Thank you.